Hello, we studied the effect of natural plant extracts on C. elegans as a means of limiting UV radiation.
Melanin is a human skin pigment, and one of its main purposes is to protect the skin against harmful UV radiation. Melanogenesis is the process in which melanin is produced, and when the skin is exposed to UV radiation, the rate of melanogenesis increases, which darkens the skin into a tan. But sometimes the skin is overexposed to UV radiation, which is when problems can start occurring.
So for some background on UV rays, ultraviolet radiation is a type of radiation that comes from the sun and other artificial sources. Although it provides vitamin D, it also has several harmful effects such as eye damage and lesions that result in skin cancer. And there are ways to prevent harmful UV impacts, such as through sunscreen. However, sunscreen only works for a very limited amount of time, and therefore an additional source of protection is helpful.
Caenorhabditis elegans, or most commonly known as C. elegans, are nematodes which have been used as model organisms for a large variety of research, especially when it comes to aging, diseases, cancer, and more. They offer the benefits of small size, short lifespan, high reproductive rate, easiness to culture, and developmental mechanisms relevant to invasion and metastasis.
The goal of the study is to contribute to research regarding UV impact prevention using plant extracts and testing the efficiency of each extract. Our hypothesis was that Olea europaea, or olive leaf extract, will exhibit the lowest amount of UV damage since the extract contains polyphenols, which is a chemical that holds multiple health benefits concerning organs. Our three independent variables were Melissa officinales, which is lemon balm extract, Garcia mangostana, which is mangosteen extract, and lastly, Olea europaea, which is olive leaf extract. These extracts have been shown to promote melanogenesis in previous studies. For example, there was a study completed by Hamid, Sarmidi, and Park in 2011 that tested the effects of mangosteen and concluded that it does promote melanogenesis to an extent.
Our dependent variable is the extent to which each extract is able to prevent UV damage on C. elegans, shown through two assays, gentle touch and osmosensation. Gentle touch assay is used to determine the C. elegans sensory input after being exposed to UV radiation. We will conduct this assay by using a probe to stroke the worm and observe how much the worm responds to the touch.
As for the osmosensation assay, osmosensation is the ability to sense a difference in osmotic pressure, which indicates the neurological health of organisms. So in this assay, we will place the treated C. elegans into a ring made of bromophenol blue and observe whether or not the behavior shows that they acknowledge or sense the ring around them. And through this test, we see how each extract prevented UV damage to the brain.
The constants within our experiment include the strength of the UV light, the amount of time the plate spent under it, the ratio of NGM to extract, the type of C. elegans, and E. coli, and the method of preservation.
Finally, the control is C. elegans cultures with NGM media only and no added extract. Now onto the methodology. First, we will prepare and preserve the E. coli and C. elegans, and we'll also set up the UV flashlight and the box. Next, we'll prepare 20 NGM plates, so 4 independent variables and 5 trials for each, and we'll add the plant extracts to the corresponding plates. We will then inoculate the plates with E. coli because this is food for the C. elegans. Then we will chunk C. elegans onto each of the 20 plates and place the plates into the cardboard box and turn on the UV light. And this marks the start of experimentation. So we will observe C. elegans and perform assays at 2 times, 0 hours and 24 hours. And at each of these 2 times, we will conduct 2 behavioral assays, which were explained before, the osmosensation assay and the gentle touch assay. And finally, we will clean up as per lab protocol and collect and analyze the data. So the statistical test we are using is a 2-way ANOVA test with replication. And we're doing this because we have 2 independent variables. One is the time after exposure to UV light, and the other is the type of plant extract incorporated into the NGM plate. And these are compared against each other to observe a potential significant difference between the C. elegans level of response and the UV light exposure. And our null and alternative hypotheses are shown on the screen here. So here are our 2 graphs showing the average response values for each assay when the gentle touch assay was performed, which is on the left, and when the osmosensation assay was performed, which is on the right. The response level of C. elegans was quantified using a scale, with 0 being no behavioral response or acknowledgement, and 3 being a huge response and a lot of acknowledgement. Here are the 2 graphs detailing the average responses at the control and the control at the baseline of 0 hours and then at 24 hours. And the standard error values for each independent variable is also shown at the bottom of the slide. So both plant extract type and hours since exposure to UV light explain a significant amount of variation in the C. elegans' response level because p-values are less than 0.001. And so some observations, our control group is significantly lower, has significantly lower response levels after being exposed to UV light compared to all the other groups. And the olive leaf extract group has the highest average response rate after being exposed to UV light. And so therefore our hypothesis is correct, and the alternative hypothesis is also correct.
As for some limitations of our experiment, our project uses C. elegans as a model, meaning that the project does not take into account bodily functions and processes outside the area being modeled. Another limitation is that it is a UV lamp being used, which has a constant and more closer exposure of light or UV rays to the plates than the sun would be. And while it is good for modeling, there are varying structures and levels of complexity between C. elegans and humans.
As for some implications of our project into society, the main point is that using herbal extracts could help individuals who are more vulnerable to UV damage, specifically those with less melanin, be more protected. And oftentimes sunscreen is inefficient in eradicating the risk of UV damage, so using extracts could give an additional boost of protection. Secondly, sunscreen only works when you apply it, but extracts are ingested, therefore they're more long lasting. And thirdly, some individuals cannot use sunscreen due to an allergy or environmental concerns, but these issues are not present with the use of extracts. As for some future aims, one of our goals is to include another variable where we test different amounts of each extract to verify the true impact of the extracts. Next, we'd like to include various other assays or ways of testing the UV impact on the C. elegans to further verify our results. And lastly, in the future, we would like to include other extracts, as there could be several other plants that can also induce melanogenesis. Here are our references. Thank you.